A good morning or afternoon to you. Thank you for joining us today. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the adaptive immune receptor repertoire community, also called the AIR community. Our speakers today, Martin Corcoran, Ayala Perez, and Oscar Rodriguez, are active members of the AIR community. Today, they will tell us about computational mining of immune receptor germline gene loci variation. Please note, this webinar is being recorded. The slides for today's webinar can be downloaded from the Materials tab in the viewer. Please add any and all questions to the Q&A box in the viewer, and those questions will be answered by the speakers. Without further ado, I'll now turn the show over to Martin Corcoran.
Thank you very much. Good afternoon, morning or evening, depending on where each of you are located. My name is Martin Corcoran. I'm a senior scientist based at the group of Gunilla Carlsson Hedestam at the Karolinska Institute in Stockholm. I'd first like to thank the organizers for giving me the opportunity to present some of our recent work and some of the ongoing work we're in development of. The Carlsson Hedestam group at the Karolinska Institute have been interested in germline inference for a number of years. This process involves the computational identification of germline immunoglobulin or TCR sequences from expressed REP-seq datasets. Essentially, the process involves the isolation of naive germline sequences that are identified from their use in multiple independent antibody or TCR rearrangements. A few years ago, we produced a software tool called IG Discover, which is designed to help people in this process. Now, the adaptive immune receptor loci are interesting for many reasons, one of which is that they're amongst the most polymorphic in the human genome. Now, when we started developing our software tool, we initially started looking at the immunoglobulin loci. So the IgK, IgH, IgL loci on chromosomes 2, 14, and 22, respectively. Now, the process that we have taken is a sort of combination of two different things. One, we tried to develop good library production techniques so that we have suitable libraries for subsequent analysis. This involves testing different types of library production, such as 5' race or 5' UTR multiplex analysis. And we eventually came to the conclusion that for consistency amongst most of our analysis, we tend to use 5' multiplex. And quite often we use two independent libraries for each sample that we're analyzing. Essentially, we take PBMCs, extract the RNA, create cDNA libraries using isotype-specific 3' UTR primers, which have a UMI embedded. We amplify using the 5' multiplex library, PCR primers, and then we sequence using the Illumina MiSeq system. This allows us to do approximately 500 to 600 nucleotide sequence reads. And these reads are fed into the IG Discover analysis program, which allows us to identify any germline genes which are present there. So this identifies both known and novel alleles. And for many of our analysis, we do subsequent genomic validation just to test the accuracy of the procedures. The overall procedure, I won't go into too much detail. You can find the detail in the Nature Communications paper we published a few years ago, and also on our website. But basically, we take what we call a starting database, which quite often would be, for example, an IMGT reference database. And we assign all of the sequences in our REP-seq library to that starting database. We look for clusters of differences and we analyze those particular clusters using a series of filters, mainly to identify the number of unique CDR3s or frequent use of J genes, and try to identify any particular features which are associated with false positives, so we can remove them from the output. So essentially that results in the replacement of the initial starting database with an individualized database, which should contain all of the functional expressed genes which are present. Now, this is just an example of one particular case where we've done this. So in this individual, when we actually did this individual initially, we had 3 novel germline sequences, but the community have been particularly accurate or active over the last few years. So those sequences have passed through the AIRR-C process and have become known alleles now. So this individual has 64 alleles. All of them are currently known alleles. So why might somebody be interested in creating individualized databases? Well, of course, it depends on what type of research you're doing. If you're interested in, for example, immunization studies, quite often it's important to get an accurate estimation of the somatic hypermutation level, both within the entire library and also within the specific antibody you might be researching. Also, lineage tracing of diverse antibodies requires us to identify the germline of the ancestral original recombinant antibody. For both of these techniques, it is essential to know all of the alleles which are present within the individual. And finally, the identification of all the gene and allele content of an individual can be used to enable association studies if you have accumulated enough cases. So I just want to give you an example here of one particular study that we used the inferral process for. So this was to use germline inferral to assemble a RepSeq suitable database for macaque studies. Now, we have been working with macaques for quite a number of years at the Carlson-Hedestam group. And when we started working on cloning antibodies, we realized that there wasn't a very good reference database for macaques. It was very difficult to actually identify accurate SHM levels. So we realized that we needed to develop a technique to build up these databases. And our original IgDiscover paper showed that there was quite a lot of diversity in just a small number of macaques that we analyzed at that time. So we decided to expand this, and we published a paper by one of our PhD students, Nestor Vasquez-Bernat, in immunity, which looked at 4 different groups of macaques. So 2 groups of rhesus macaques, the Indian and Chinese origin macaques, and 2 groups of cynomolgus macaques, the Indonesian and Mauritian cynomolgus macaques. Now, these 4 groups are widely used in immunological studies in different parts of the world. So it was quite important that for us at least, to create a reference database that many people could refer to if they wanted to get accurate assignment of any antibodies they identified. And we used essentially the same process, isolating blood samples, creating total RNA, and doing cDNA synthesis of 2 independent library sets, one with the primers in the 5' UTR and one with them in the leaders region. We analyzed these using the IgDiscover software tool, and we were able to combine all of the alleles in a dendrogram for the different groups. This is an example of the output from the 27. What you see here is a dendrogram of the IgHV alleles from the 27 macaques. Now the red spokes on this wheel here are actually identical sequences from the starting database that we used, which was the Sorelli et al. Macaque database. From a paper published a couple of years previously. Now, what is sort of interesting to us when we looked at this is not the fact that we don't see too many of the Sorelli alleles present in our database. We do see quite a lot of them, but not all of them. It's the fact that we actually see large gaps in different regions. So to us, this was interesting because it highlighted two different possible explanations. One is that the assembly methods at the moment may be missing out parts of the IGH locus in some of these animals. And the IGH locus is, of course, very complex, and I'm sure Oscar will provide some details of this in his talk. But we also know that the IGH region is subject to deletion in different individuals. And we were able to identify quite a number of what we are determining as IGH genes in macaque, which are present in some animals, but deleted in others. Now, this is actually quite similar to the copy number variation situation that you see in humans. And also, we wanted to actually check to see whether this was a regular feature in macaques. And of all of the animals we tested, we actually noticed this type of deletion in all of the animals, but for different genes. So this indicates that allelic diversity is very common in the animals, but also structural variation is common as well. Now, what this suggests to us is that if you're intending to create a reference database of IGH V genes, for example, using an outbred animal, you may find that these animals have these structural variations. And if you use, for example, a haploid assembly, even if it is a very accurate, completely well-sequenced assembly, you may find that it is missing quite a number of genes. So in order to create a more comprehensive database suitable for RepSeq assignment analysis, it is probably essential that you use multiple animals. Now, the inferred germline database that we have done, we think is highly comprehensive in terms of the coverage of germline sequences. It certainly not including all of the allelic sequences, but we think it includes representatives of all of the genes which are present in macaques. However, because we are doing this kind of agnostically in terms of the gene nomenclature, this means that we still have some difficulties in terms of determining whether a particular germline sequence is associated with a particular gene. So this will probably require the combination of genomic with RepSeq analysis in future to solve this problem. Okay, so that's something that we've done quite recently. So I want to sort of talk about some of the ongoing development of the program. So there is an IG Discover updated version, which we're going to release this spring, which has a number of interesting features which are in development at the moment. First, it's MAIR compliant, which means that it has an output now which will be possible to integrate with other MAIR compliant software tools. We have greatly improved the TCR discovery part of the program. We had a TCR discovery section for a number of years in the program, but we realized that it did require a number of changes. It was basically using the same procedures that we were using for IGH analysis, and there are some differences with TCRs that required some coding differences. It has a greatly improved haplotyping facility, and we has what we call a CoreCant genotype module, which allows us to do much more rapid genotyping from our analysis set. We've also improved the J and D discovery part of the program. So in terms of TCR analysis, we wanted to be able to analyze all four isotypes, the TRG, TRB, and the overlapping TR alpha and delta isotypes. So in order to do this, we first developed a good library production technique. So we applied our previous experience and developed a 5' multiplex library primer set for the alpha, beta, gamma, and delta TCR chains. And we were able to get very good coverage now of the full VJ or VDJ recombinants. We apply these to the computational analysis of the updated IG Discover tool, and we were able to identify both known and novel allelic variants within the libraries. We then apply the core count for genotypic validation. Now, in terms of like TCR identification, we have some specific changes that we find are required in contrast to the IG analysis. The program itself needed an updated CDR3 detection. The previous CDR3 detection system did not work very efficiently with all the TCR chains. So we have greatly improved that within the program. So now you get a much higher number of sequences which can be analyzed from your library. We have also used some of the updated validation techniques, such as PCR Sanger sequencing and haplotype analysis, to go through a lot of the alleles that we are finding, just to make sure that the program is giving us accurate outputs. So just to give you an example here of an analysis from one of the libraries we produced. This is validation by inferred haplotype with IG Discover. So this essentially uses the fact that if we have heterozygous anchors, so in this case this will be heterozygous J genes, these particular J alleles will only recombine locally. So they only recombine with V alleles which are present on the same chromosomal region. And that allows us to do computational analysis of the counts we get for the different V alleles associated with the J alleles. This particular picture here shows one individual who has a total of 19 novel V alleles present in their repertoire. Also, the program allowed us to identify novel J allele in this individual. Here's a novel TRA-J44. And the fact that you can actually identify novel J alleles allows you to do this haplotype analysis of the V alleles. And just to show the accuracy of this, we did a familial study as well, where we analyzed a father, a mother, and a child. And in this case, the child inherited 9 novel alleles from the parents, 7 of these from the father, and 4 from the mother, and 2 of which were inherited homozygously. Now, both of these analyses indicate that once we start looking at the TCR loci, we're going to find quite a lot of variants present. So these are just a few cases, but we are finding multiple novel alleles the more samples we are examining. And I expect that the more cases that are passed through this type of analysis, the better chance that we will get a more comprehensive database. It's clear at the moment that the current TCR database is greatly underestimating the number of alleles which are present in the human population. And certainly our results are consistent with what people are finding in general genomic analysis, which is that the greatest degree of variation that you will see is in African populations, who clearly have not been studied to sufficient depth to identify alleles. But we're also finding population-specific alleles from other populations around the world. Now, finally, I just want to go through the genotypic module that we are developing at the moment. Generally, when you are thinking about a genotype of an individual identified through RepSeq analysis, in the past, this was usually just the output you would get from a program like IgBlast or VQuest. Now, of course, these particular analysis tools are dependent on having good databases, so they don't really identify novel sequences. And this has been supplemented by subsequent programs which can identify novel alleles present. However, neither of these really actually give the full picture of a complete genotype. And we realized that we were missing quite a lot of the low frequency genes and alleles. Now, for example, in the IGH locus, if you analyze multiple individuals, you tend to find that in practically all individuals, the same sets of genes are present at high frequency. And the same way, another set of genes are present at pretty low frequency. And a lot of the tools that we're using at the moment for individual database analysis have the problem that in order to actually get high quality results, we need to apply very high stringency to the analysis. So we wanted to develop a tool which would work both rapidly and give us high quality analysis. So we developed a technique called the co-count analysis module. Now, this is based on a very simple principle, which is that most recombined VDJ sequences, they don't actually contain a full-length VDs or Js. However, they will contain a core of these. So usually there's a small amount of sequence which is missing at the recombination site. So we created what we call a modified core database, which allows much more highly accurate identification of even low frequency genes and alleles. Now, what this does is that we actually only remove a very small amount of sequence from each of these to create the core database. But that is enough to actually give us very high quality output. Okay, so the requirements are a comprehensive database and a RepSeq library with sufficient unmutated sequences. So they work very well from IgM, IgD, Kappa Lambda, and all the TCR libraries. It doesn't work for IgG libraries, so that is a limitation. Now, you require high quality sequence libraries. So with our MySeq 2x300 V3 library sets, we have to avoid high cluster density. But if you do that, then you actually get very good quality outputs from this. And you don't need very large counts to actually get a good genotypic output. Now, in order to test this, we realized that we're going to have to analyze a known genotype against a expressed sequence. So we identified a high gene content individual. So this is an individual who has several structural variants present. So the V18 and V39 structural variant, also the 510-1 and 364D variant. The individual has several genes which are missing in multiple individuals in the population, 330-3, for example, or 169-2. So the individual has quite a lot of genes, so that gives us a good starting point. So what we did was we amplified every single one of those genes. So 50 IGHV genes, 64 alleles to be identified, 27 IGHD genes, and 6 IGHJ genes, which 7 alleles in total. So design primers to amplify all the expressed sequences. These were all cloned into plasmids and subsequently Sanger sequenced. So all the expressed V, D, and J genes were targeted in the individual. And this was done by one of our PhD students in our group, Sanjana Narang. Now, the result of this was that the Sanger sequenced genotype showed 100% identity to the core count genotypic output in this individual. So we had 100% identity. So just a quick summary of this, that the core count genotyping is performed on the filtered output of IG Discover. Batch analysis is extremely rapid. We can do approximately 200 genotypes in less than 2 hours. Filtering is simplified. We use an allelic ratio expected frequency for the genes and alleles, and CD4 diversity filter. We get highly accurate genotyping of low-frequency genes, and most importantly, it's very good at discriminating between alleles which have 3' end variation. This can be quite important because normal assignment methods often ignore the 3' end variation. And if you have a 3' end variation in a receptor gene, quite often this variation occurs within the CD4. So it might be important for functional. So just to conclude the talk here, I just want to emphasize that we're finding that diversity is extensive in outbred species. And this needs to be taken into account when you're doing computational analysis. The diversity involves both gene and allele content. And probably this will require the combination of RepSeq and genomic methods to really define diversity. Databases will become more comprehensive and accurate as this work continues, and the availability of these more comprehensive databases will allow the development of high throughput, rapid and consistent methods that will enable future association studies to be performed. Now, I just want to acknowledge the members of the Carlson Hedestam group that have been involved in some of this work. So, as I mentioned, Sanjana was involved in all of that validation cloning. Matthias Kaduk and Mark Chernyshev are involved in the software development and coding in the group. Marco Mandelesi is involved in the TCR analysis. And we work with Christopher Sundling and Anna Farnet at the Karolinska, who provided some of the samples that we are analyzing. And I'd finally just want to thank our funders as well. Thank you.
Hi, everyone. My name is Allelic. I'm from Guriari's lab. And today I'm going to talk about the germline variation that we can see from both T cell and B cell from analyzing AIRR-C data. I'm going to continue on from what Martin presented before. So when we analyze B cell and T cell receptors, what we look for is often the germline variation. And the germline variation of the adaptive immune system can be actually divided into two. One is the genetic side, and one is the environmental side. The genetic side, as you guys know, it's mostly connected to the connected to the combination of the V, the D, and the J. And the environmental side is what we encounter what happens when we encounter a pathogen, somatic hypermutation, and class switching, and so on. And when we want to analyze the AIRR-C data, what we want to do is sort of like a play on a backward engineering. So we start from B or T cell receptors. We take those receptors and we sequence them using any of the platforms. And then what we want to do is align the sequences using either IgBlast, Sparties, or even IMGT, and get the V, the D, and the J calls. We want to get these calls because we want to know what an individual has, and then we want to infer from this the different variation. The different variation that we can see is, first of all, as discovering undocumented allele candidates. So what I mean by undocumented alleles is that we are starting from a germline set that is often taken from IMGT. Now, this germline set doesn't include the entire world. It's just what we know currently. But as we sequence more and more individuals, we use different tools to discover a new variation of alleles. And we can use either of the tools of TIGR or IG Discover, as Martin said, or even parties to discover this novel variation. And this adds on to the germline variations that we can see. Then when we go downstream, what we want to do is infer the genotype. Inferring the genotype gives us sort of like a snapshot of the genes and the allele one individual is carrying. As we can see from the picture below, what we're seeing is that we started from the receptors, we aligned the sequences, and then for each individual, we got the different gene and allele, the top row and the bottom row for each individual. From this, we can start to see the different presence of all of the different combination of the genes and alleles. And then we can go on to further downstream analysis. The downstream analysis to get some more variation can be either discovering deletion, as you can see from the figure below that are marked with an X. It could be maybe looking at the upstream sequences. When we sequence, sometimes we sequence also the variation of the 5' UTR, the untranslated region. We can also look for allele biases when we look more closely into the gene usage. And lastly, we can look at the haplotypes, meaning phasing the two chromosomes by using either an anchor gene or another method, and then seeing which alleles and genes are sitting on each of the chromosomes. So basically using this inference steps, we've done a few studies that look into the different germline variation. The first thing that we looked at is discovering undocumented alleles. So we published three different papers that went on to discover undocumented alleles for both the heavy and the light chain for B cells and the TRB chain for T cells. So as you can see, we know quite a few. Let's take, for example, the IgH. We know 287 known alleles. But with the paper that we published, we discovered 25 more undocumented alleles. And in our latest publication for the T cell, from 64 known alleles, for V genes, we discovered 38 additional novel alleles. And this is just part of it. But when we talk about genomic variation, some of it is going into the genotype, is knowing the influence of one gene on the usage of others. In our latest publication of the T cell, we looked more closely into the genotype and specifically the TRBD2. So for T cell, for the beta chain of the T cell, there are two Ds. There are D1 and D2. Now, D1 can only recombine with J1 genes, and the D2 can only recombine with J2 genes. The D2 holds two alleles, allele 01 and allele 02, which are differentiated by one nucleotide. It's between a G and an A. Now, this small little snip that differentiate between the two alleles can highly influence the usage of other genes that an individual carry. So what we did is we took a cohort of around 700 individuals that were sequenced for their beta chain of the T cell. And we genotyped and we wanted to see the different combination that the individual has of the TRBD gene. So what you see in the figure here is the y-axis is the usage of the gene, and the x-axis are different genes. And the color corresponds to the genotype of the TRBD2 gene. And what we saw here that was very, very interesting is that if we're homozygous to allele 02, then we're more prone to use the J2 genes and less the J1 genes. But if we're homozygous to allele 01, which is marked in red, then we're using the J2 gene much less. And this was very interesting discovery for us, that there is a strong bias between the genotype of TRBD2 and the usage of other gene, which is an incentive to further going into genotyping AIRR-C data to understanding the different influence of one gene on another. Another thing that we developed in our lab is a tool for haplotype inference. So this tool for haplotype inference was published back in 2019. The name of it is Rabbit, which is an R antibody haplotype inference tool. Basically, in this tool, what we did is we took repertoires of heavy chain, and we looked for one gene that was heterozygous that can be used as an anchor to phase the two chromosomes and to show us how the gene and their alleles are sitting on each. So what we did is we detected the J6 as heterozygous and is shown as heterozygous in a lot of individuals. It has both allele 02 and allele 03. This helped us to see the different recombination that this gene is having with different V and Ds. And from there, we could utilize a Bayesian method to phase a chromosome and basically realize on which chromosome each of the alleles is sitting. From this project, we also figure out that by phasing the chromosome, we can also see if one of the genes is missing on either of the chromosome, what we call a deletion. So to infer deletion, what we did is we phased the chromosomes and look for the spots where we don't see any recombination on one of the chromosomes. By using the Bayesian method, we could get a confidence level in our call. This confidence level help us understand if there is helps us understand how confident are we in the inference that we've made, and from this to understand if there is a deletion on one chromosome. So in the top figure on the right side, we developed a method to allow us to infer double chromosome deletion. And on the bottom one, utilizing the haplotype method, we were able to infer single chromosome deletion, which means that on one side we have the present gene, and on the other side we have a deletion. Lastly, the tool also offered to show the complete haplotype of an individuals, where we see the different alleles sitting on each side of the chromosomes. So we developed this tool for B cells, but just this last year we adapted this tool also for T cells. We adapted this tool for T cells by utilizing the TRBJ16 as an anchor. This gene is heterozygous for a lot of the individuals. And from this, we could discover different allele patterns and association between the genes of the TRBV. So here on the right, what we see is each row is a different individual, and each column is a different gene. And the colors are coded to the different allele. Now, the top panel is showing the chromosome for TRBJ16 allele 01, and the bottom panel is showing the TRBJ16 allele 02. And what was most interesting for us is to actually see a linkage between two V genes. So we saw a linkage between V72 and V43. And what we saw is that every time that an individual is carrying allele 02 in TRBV72, then there is a deletion in TRBV43. Now, we saw this linkage on both chromosomes, so it's not limited to one of the sides, and it's present on both, which was very interesting to us. And when we believe that if we do more studies and we infer more haplotypes, later on we can maybe associate this linkage to different diseases or to other germline variations. And all the things that I've showed you up until now, we took all the data from the different projects of both the heavy chain and the light chain and developed a site called VDJbase.org. Now, this site holds a publicly available T-cell and B-cell database, and this allows you to see the different genomic variation by producing different reports to compare between different individuals, samples, both the genotype, the haplotype, allele usage, and many, many more. And it's very easy to use and allow you to search for different projects and individuals. Now, this project, the VDJbase, is available online, but you can also install it locally on your computer and add your own privately available data sets. And further, to pre-process your data set the same way as we do in VDJbase, we created a Docker version of our processing pipeline, which is available in Docker Hub. So now I'm going to do a very short demonstration of our site and the different reports that we can produce. So this is our site of VDJbase. This is the landing page where you start in VDJbase. From here, you can go into the different tabs. On the most right side, you have the Help tab. The Help tab has a user guide, and the user guide has videos to show you all about the website and how you can access all the different things. In the AIRR-C data tab, we can go to Explore AIRR-C data. This tab is showing us the different projects and the different chains that are currently available on the site. So for the heavy chain, for example, we have around 600 samples that are coming from 425 individuals. And we can see down below in the table the different projects that we have, their accession and ID from which institute they came from, the number of subjects, sample, and so on. And here in the figures, we can also see the division of the samples to the different clinical status, the cell types, and the tissue. Now, if we go here, we can switch between the heavy chain. We can unselect the heavy chain, and we can see what we have, for example, for the T-cell. So for the T-cell, currently we have 6 different projects that are coming from different library preparation, from both 10x genomic, full sequencing, even partial sequencing using biomed primers and adaptive. And we have a very nice division between different clinical status, either CMV or healthy. And we have a very nice division between male and female. Now, when we want to produce reports, we can go into the Samples tab. The Samples tab show us the different samples that we have. And for each one, we can get the different metadata, meaning the status of the sample. Here we can see from which study it came from, the sequencing protocol that was used, the information about the subject that we have, which inference pipeline was used to produce this sample, and the tissue processing for this specific sample. For each sample, we have a few columns that give us a little bit more information. We have the number of reads that were assigned to this sample, the tissue, if the sample was sequenced using UMIs, unique molecular identifier, if the sequence is full or partial, meaning if it was sequenced from the start of the V until the end of the J, or if it was sequenced with biomed primers, meaning it was starting from the middle of the V. We can also get some information about the genotype. Here we can produce the genotype reports, both the reports that were done by Tiger and the genotype analysis, and OGR-B statistic. And if the sample is haplotypable, we can also download the haplotype by clicking here. And this allows us to show the haplotype analysis. For the reports, we need to go into the report tab that is here on the third tab. And we have different reports that we can produce. We can produce a genotype list, a genotype heat map, or a haplotype. And we can also see allele appearance and heterozygosity. These two allow us to see what alleles are more common in the data set that we have, and which genes are heterozygous. And we can also get a comparison of the gene frequencies. So to produce a report, we go again into the samples tab. We go into under the name and we select a few samples. We can select, let's say, 3 samples that we want. We'll select these 3 samples and we'll filter them out. We go again to the reports, and then we can choose whatever report that we want to produce and compare. We can produce a genotype list. This window pops up and allow us to further organize our plot. So we want to show the graph for each of the samples. We can sort the gene either by an alphabetical order or by a locus order. And we can also select if we want to see some of the genes or all of the genes here in the 2 tabs below. And then we just click on Run the Report. So the report is going to run on a different tab, but now I'm going to show you a few reports that I've already produced. So one of the reports is heterozygosity. In this report, we get, this is done for the heavy chain, we get the heterozygosity for the V, the D, and the J genes. And once we go on one of the genes, we get this pop-up window that shows us the heterozygosity, in this case, for the V, for IGHV 118. In this case, we selected 4 individuals, and we see that 2 individuals are heterozygous for the gene, and 2 individuals are homozygous for this gene. And this can be done for from 1 individual into the entire cohort for each of the chains. Another report that we can produce is a comparative heat map of genotype. So in this report, what we see is each row is a different individual, and each column corresponds to a different allele, and each row is a different individual, and each column is a different gene, and each column corresponds to a different allele. And this is, again, an interactive plot where we go on each of the box, we can see the name of the individual, the gene, the allele, and the KDIF value. Now, the KDIF value come from Tiger's analysis, whereas the confidence, it's the confidence value for the inference of this genotype call. In this report, it allows us to basically compare the different genes, the different individuals, and see the different variation that we can discover from the genotype. I mean, if we go for V118, we can see these 2 individuals are heterozygous for this gene, while the 2 other individuals are homozygous for the gene, the same as the heterozygosity plot. And in the cases where we have a deletion, as I said, that we can infer from the haplotype, we're seeing here that we have this deletion that is marked in dark gray. Another report that we can see is comparative of the gene usage. Now, this report is not interactive, and it's downloaded as a PDF. This report allows us to compare between different individuals. Each dot is a different individual that we have. The y-axis is the usage of the gene, and the x-axis are the different genes. And we can compare between them all. If we go back to the plot that we produced, so here are the 3 different individuals from the T-cell cohort. The y-axis are showing us the different genes, and the color is coded into the different allele. And here we can compare. So if we take D2, for example, which we know could be heterozygous, we see that for this individual, is carrying both of the alleles, while the other D is showing just, for this individual, is homozygous. So These are the reports, and as I said, all of this is available to download and is also publicly available both on GitHub and in Docker Hub. For the future plans of what we want to do more with VDJbase is we want to adapt our pipelines to other species. We want to add more subject to more projects and more individuals to VDJbase. And we want to improve the visualization of the site and also allow to other people to contribute with different suggestions for more comparative visualization between the projects. And this is it. Thank you for listening, and I want to acknowledge all the groups that were helping us to produce all of the results. So thank you.
Hi, everyone. My name is Oscar Rodriguez. I'm a postdoctoral fellow in Corey Watson's lab in the University of Louisville School of Medicine. And today I'll be talking about detecting genetic variation in the immunoglobulin heavy chain locus using targeted long-read sequencing. This is the outline of the presentation. First, I'll go over genetic variation that has been previously found in the immunoglobulin heavy chain locus, or IGH, and the advantages of long-read sequencing over short-read sequencing. Then I'll go into the framework that we developed to resolve the IGH locus using long-read sequencing in a high-throughput fashion, and how we're applying it to a cohort with adaptive immune repertoire sequencing data, and end with applications to the TCR loci and the IG loci in rhesus macaque. So genetic variation is defined by any genetic difference between individuals. In this case, one individual has a G and the other a C. And genetic variation can range in single base pair differences to small insertions and deletions or indels, to much larger genetic variants, such as structural variants or large insertions and deletions. And all of these genetic variants have been observed in IGH. In humans, the IGH locus is located at the telomeric end of chromosome 14. And as we know, it contains 4 different sets of genes, the variable, diversity, joining, and constant genes that come together to form the heavy chain of an antibody. And the selection of different genes produces a diverse set of antibodies capable of binding to a large number of antigens. At a population level, there also exists haplotype diversity. 2 individuals with the same gene can have different alleles. And this contributes to the overall diversity of the repertoire. Current data shows that there's a large amount of allelic diversity in the population. Here I'm showing different variable genes in IGH and the number of alleles per gene. The first V gene in IGH, V6-1, contains 3 different alleles that differ by 1 to 3 bases. In 2013, when the IGH locus was sequenced for the second time, a large number of structural variants were identified. Here I'm showing the IGH locus and the structural variants identified just in a single individual. When individuals from different populations were screened for structural variants, several additional structural variants were identified. These include insertions, such as a 46.6 kb insertion that introduces 3 genes into the locus, genes 169D, 169-2, and 170D. Large deletions, such as the highlighted 32.7 kb deletion, and other complex structural variants. In addition to the large amount of structural variants, there's also an extensive amount of repetitive sequence. The second row of each panel in this IGH locus schematic from Matsud et al. Shows regions that are highly identical to each other. For example, all the red regions contain sequences that are highly similar. Additionally, there are different repetitive elements, such as a loop, lines, and simple repeats. The structural variants and repetitive sequence has made detecting genetic variation in IGH a very hard problem, especially with short-read sequencing and next-generation technologies. And this has to do with the read length that they produce, which ranges from 50 to 250 base pairs, depending on the platform. A typical workflow begins with aligning reads to a reference genome. And the difference between the generated reads and the reference genome corresponds to genetic variants. However, this methodology represents a limitation. For example, if the input DNA contained repetitive or identical sequence, then the generated reads will also contain repetitive or identical sequence. and proper placement of these reads on the reference genome is often difficult because they could be put in different locations. And if the reference genome is incomplete, it could lead to misalignment of reads, and ultimately genetic variants will be harder to detect. One way to overcome this limitation is to use longer reads that completely span genetic variants and contain additional unique sequence. Current DNA sequencing platforms that are capable of generating long reads are the PacBio and Nanopore platforms. For this talk, I'll only be talking about the PacBio platform, but our methods are applicable to Nanopore as well. On the DNA sequencing PacBio platform, half of the reads are greater than 30 kb for a typical whole genome sequencing run. In this example, the longer reads will identify the insertion that is missed by short reads. In studies that have used longer reads across the whole genome, a large number of novel structural variants have been identified. In one of our previous studies, we compared structural variants identified by different sequencing technologies. The plot here is color-coded by which technology detected the structural variants. On the right are insertions, and on the left are deletions. And so as you can see, the majority of the structural variants detected were detected just by PacBio. And we concluded that 83% of insertions were missed by short read data, and 3 to 7-fold more genetic variation was detected using long read sequencing data. So there's multiple pieces of evidence on why genetic variation, both coding and not coding regions, matter in IGH. Twin studies have shown that the antibody repertoire gene usage within monozygotic twins are more similar than in unrelated individuals. Functional studies have shown that specific genotypes within the IGH locus correlate with specific gene usage of specific genes. And genome-wide association studies have identified IGH polymorphisms with different diseases and traits. So given the complexity of the IGH locus, the amount of structural variants previously found, and the limitations of previous DNA sequencing studies, and studies pointing to a genetic effect on the antibody repertoire, we sought to develop a framework that was capable of resolving genetic variants in IGH, such that they could be used for future genetic antibody repertoire studies. In 2020, we published a framework on how to resolve the IGH locus in a high throughput and scalable fashion using long read sequencing. We did this by targeting the IGH locus. DNA probes were designed to specifically pull down DNA from IGH. This DNA was then sequenced using long read sequencing and analyzed with a framework that we developed called iGenotyper. The input to iGenotyper are CCS reads, or what is now being called hi-fi reads. These long reads that we generate are on average 6 kb long and have a read accuracy ranging from 98.9 to 99%. The first step in iGenotyper is detecting phase SNPs from the CCS reads. This is done by aligning the reads to the reference genome, or IGH locus in this case, and detecting SNVs. These SNVs are then phased using WhatsApp. Reads that are overlapping multiple SNPs are used to determine which SNV alleles occur on the same haplotype. The phased heterozygous SNPs allow us to partition the reads into their respective haplotype. For example, here, the reads with bases G, T, G, A, C all belong to haplotype 1. And so by using this information, we could partition the sequence PacBio reads into a set that's haplotype 1 specific or haplotype 2 specific. And this is done using a probabilistic framework where the quality read base score of the overlapping SNP position is taken into account. And so after the reads are partitioned into each haplotype is assembled using their respective haplotype-specific reads. And then these assemblies, these locus-wide assemblies, are then used to detect genetic variants across the whole locus, genetic variants such as SNPs, indels, and structural variants. Indels and structural variants that are resolved within the assemblies are detected using a multiple sequence alignment and a hidden Markov model that models different structural variant events. Additionally, alleles are extracted from the assemblies and compared to the IMGT database to annotate which alleles are present in the sequence sample. So we first tested our method on a ground truth sample. We captured the IGH locus at an extremely high coverage. 95% of the IGH contained more than 70x coverage with CCS reads and more than 940x coverage with raw reads. And importantly, all the genes were sequenced at a high coverage. However, one thing to note is that these results are from a much earlier PacBio platform. I'll present much more current data later on, showing that we're actually getting much more coverage. And so with the previously generated sequencing data, the IGH locus was assembled and compared to a ground truth. And so we were able to recapitulate 98.7% of the IGH locus to an accuracy greater than 99.99%. And the alleles that were extracted from the assembly were all correctly annotated. We also initially applied our capture method and iGenotyper to 2 diploid samples from the 1000 Genome Project, NA18278, which is a European representing sample, and NA19240, which is an African sample. On the top is the IGH locus with different variable genes, and on the bottom is a schematic showing the haplotypes for each sample. The gold haplotype is the maternal haplotype, and the green haplotype is the paternal haplotype. And what could clearly be seen is the large number of structural variants that are present in these samples. And some of these structural variants are relatively large, such as the 74 kb and 61 kb deletions. The structural variants that are boxed off are novel structural variants that were identified in these 2 samples. Another pattern that has been previously observed is the structural variant difference between samples. For example, NA12878 is homozygous for the 60.9 kb deletion, while NA19240 contains the reference sequence, which harbors 4 different genes. And the genes for these 2 samples were also genotyped. The white boxes with diamonds are genes that are deleted. NA12878, the European sample, has 13 genes that are homozygously deleted, as opposed to NA19240, the African sample, that has no genes deleted from both haplotypes. Or no genes are deleted from both haplotypes. Or there are no genes that are deleted from both haplotypes, sorry. And additionally, we identified 16 novel alleles that in just these 2 samples that were not present in the IMDT allele database. And so one thing that should be noted is the allelic diversity that exists between these 2 samples. For example, IGHV 169 contains 4 different alleles across 4 haplotypes. The assemblies that were generated were validated with 3 orthogonal methods, and all methods showed that the assemblies were greater than 99.99% accurate. The accuracy of the SNPs detected by iGenotyper were also tested. The gold color represents false positive SNPs, the purple are false negative SNPs, and the blue are true SNPs. In total, iGenotyper detects around 3,000 SNPs for this sample, of which 99% are true SNPs and 46 are false positive SNPs. The same analysis was done to SNPs detected using short-read data from the same sample. Around 4,400 SNPs were detected, of which only 49% were only true SNPs, and about 2,200 were false positive SNPs. SNPs from short-read data and genotyping arrays from the 2 1000 genome samples, NA19240 and NA12878, were also compared to SNPs identified by iGenotyper. In NA19240, the SNPs from the short-read data only contain 58% of the SNPs identified through our framework. In NA12878, the SNPs from short-read data contained only 76% of the SNPs identified by iGenotyper. And the majority of the SNPs that were missed by short-read data and identified just by iGenotyper were within IGH regions that were inaccessible to short-read data. We also went on to determine the effect of sequencing multiple samples in a single sequencing run. And so we tested 5 different sequencing runs. They contained 4, 8, 16, 24, and 40 samples. So as expected, the coverage of each sample decreases as more samples are sequenced at a single time. And this leads to a decrease in the number of SNPs identified. However, the false positive rate, or the number of false SNPs identified, stays consistent, which told us that we could apply our framework to large cohorts. So iGenotyper outputs several files that are typically used in genomic studies and could be used by other tools for other downstream analysis. There are FASTA files with the allele and assembly sequence. There are BAM files that contain alignment of CCS reads and the assembly to the reference. There are VCF files and beta files that contain the SNPs, indels, and structural variants, and the allele calls. Additionally, statistics and plots are generated for every single sequencing run. Okay, so now that I presented the framework that we developed, I'm going to move on to studies where we're deploying this framework. In one study, we generated paired genetic iGenotyper and antibody repertoire data for 154 individuals in order to study the overall genetic effect on the repertoire. For this talk, I'll focus just on the output and results from our long-read sequencing, capture, and iGenotyper method. So in this cohort, different samples were sequenced using different PacBio sequencing platforms. Sequel 2 is the latest platform, and RS2 is one of the earlier PacBio platforms. The y-axis is the locus-wide coverage, and the x-axis are different sequencing runs. On the Sequel and Sequel 2 platforms, we multiplexed and sequenced multiple samples per run. And across all the samples, the mean coverage was 76x. One of the biggest improvements platform-wise was the gain in accuracy of the sequencing reads. For Sequel 2, we're generating reads that are on average greater than 99.98% accurate. And so this overcomes the historical fact that long-read sequencing used to have lower accuracies, but now with the latest platform, the accuracy, like I said, is greater than 99.98%. It's highly accurate. And so this allows for downstream validation of results obtained from the assemblies and for much higher quality assemblies. So across our whole cohort, we identified more than 20,000 SNPs. And so given the complexity of the locus, we hypothesized that many of these SNPs will be novel. And so in order to check out this hypothesis, we compared against a database called dbSNPs, which contains SNPs that were identified from huge genomic studies. And we found that 23% of the SNPs that we identified in IGH were absent from this database. Overlapping the SNPs with genes, we observed that many overlap different gene components. Interestingly, when this is viewed at a gene level, not all genes contain the same number of SNPs. For example, IGHV 439 contains more SNPs in LPAR1 than any other gene. IGHV 4-30-2 has the most SNPs within the gene allele coding sequence. IGHV 3-23 has the most SNPs in its introns, and V321 has the most SNPs in its RSS sequence. And so as I talked about in the beginning of the presentation, genetic variation ranges from SNPs to indels and structural variants. And so similar to previous findings, we also found additional novel structural variants that affect the number of genes in the locus. The structural variation loci track shows all the regions in IGH affected by structural variants. The SV track shows all the SVs that we found in this cohort. And the bottom track, this track shows all the 53 out of 90 genes that are not deleted by structural variants. And so one important thing to note here is that this is from a cohort of only 153 studies. What will probably end up happening is similar to other genetic studies. When the cohorts are much larger, we'll probably end up finding much more genetic variation. And so here's a zoomed in figure of the multi-copy number variant that we identified that contains 3 structural variation alleles. One of the structural variation allele is a 259 kb deletion that deletes 14 variable genes. And the other is a 284 kb deletion that deletes 16 variable genes. What's further interesting is that these deletions were only observed in white individuals. And this is likely explained by the fact that this structural variation is mediated by another structural variation that occurs at a high frequency in European populations. We also found a large deletion in the IGHD region that has been previously observed in AIRR-C data. this 9.6 kb deletion removes 6 out of the 26 IGHD genes of the locus. And interestingly, this deletion is common. It's present in 23 out of 76 individuals, and it's homozygous in 5 individuals. So here's one individual who is homozygous for the IGH deletion and heterozygous for the 284 kb deletion in the IGHV region. So in combination with the other structural variants identified just in this individual, 13 genes are deleted from both chromosomes and 22 genes are deleted just from a single chromosome. So in addition to structural variants that affect gene copy number, we also identified an extensive amount of indels and structural variants that are in the non-coding regions. For example, some of the indels were expansions of tandem repeats. Here is a polymorphic expansion of a T homopolymer. So the left is a screenshot of assemblies from different samples that were generated, and on the right is a screenshot of raw reads. And so what you could see, for example, one individual has 8 T's deleted, while others have an increase of T's. And all of these indels are supported by raw reads. So in this cohort, the number of T's in this region ranged from 16 to 50, with the majority of individuals having 23 copy, although there does seem to be a bimodal distribution. Another example includes an expansion of contraction of larger tandem repeats. Here's a VNTR that has an 86 base pair motif that's 3 kb away from IGHV 3-20. Almost all individuals in the cohort contain 4 copies of this 86 base pair motif, but 15 individuals contain 8 or more copies. Other structural variation events are even more complex. Here's a 4 kb region upstream of IGHV 1-3, where 3 different sequences were found that differ by SNPs, insertions, and deletions, indicative by the red tick marks and gaps. An in-depth analysis found that each sequence contains a distinct but similar expansion of a 21 base pair motif. However, each sequence contains a different copy number of the 21 base pair motif. And the copies of the motif within each sequence do not necessarily have a high sequence identity. And this is likely indicative of a polymorphic tandem repeat expansion that has accumulated mutations. What makes this further interesting is that the frequency of each sequence is found at different proportions across populations. So all these indels and structural variation examples, they show the strength of completely resolving haplotypes as structural variation sequences. And so to conclude, we also identified a large number of novel alleles. On the left is a pie chart of all the IMGT alleles and the novel alleles that were identified in this cohort. The individuals with the most novel alleles, self-reported as Black or African American, indicative that common alleles are in less studied populations are missing. So we also applied our framework to the TCR loci. And so all the generated data and results that I just presented for the IGH locus, we also generated for the TCR loci. And so here I'm just going to briefly show some results. Capture and long-read sequencing for several 1,000 genome samples was performed and analyzed using iGenotyper. The TRB haplotype assemblies for 2 of the 1,000 genome samples is shown here. For the bottom sample, we completely resolved the TRB locus, and we identified 2 20 kb insertions, and both of these insertions contain 4 genes. The TRA and TRD loci were also completely haplotype resolved, and again, this means that there was an assembly that completely spanned the whole locus. And so similar to IGH, we use these assemblies to identify SNPs, indels, and structural variants, and to genotype the genes that are present in these loci. Recently, we started extending our methods to resolve the IG loci in the IG loci of Rees-McCock, and this is in collaboration with Melissa Smith and Steve Bosinger. So currently iGenotyper has functionality to work with the rhesus data, and preliminary results show that we're effectively sequencing the IG loci. So here are sequencing reads in the kappa locus for 4 out of 34 recent macaques that we recently captured and sequenced. And so just from the sequencing read coverage profile over the IgK loci, we're clearly seeing large structural variants. If we zoom in, we also observe a large number of SNPs in just these 4 individuals, indicative by the colored tick marks. So to conclude, we developed a long-read sequencing targeted approach along with a bioinformatic tool called iGenotyper, and used this framework to resolve a large amount of novel genetic variation and alleles in the Ig and TCR loci. And we're currently extending this method to the rhesus macaque. And to conclude, I'd like to thank everyone who took part in all these studies. Thank you.
All right, well, thank you very much, all 3 of you, for giving really wonderful presentations. I'm going to allow just a minute or so if people want to think about any additional questions that they may have thought of, and just to impose a little bit on Martin. Several questions were asked, and you already responded to them through the Q&A box, but for the convenience of people who will be watching this later on demand, could you just say a couple of words to answer the questions? And 2 of them, starting with the first one, which is, to which extent do you think reads length can affect the inference? For example, 2 times 250 base pairs for TCR-AB.
Sure. What I would say about that is, of course, if you want to do a accurate inference of a full-length VDJ sequence, it's best not to use primers which actually overlap part of that VDJ. So, for example, framework 1 primers or primers which are present within the J region, you essentially cannot trust the sequence which the primer overlaps. So you can only trust the sequence which is internal to the outer and inner primers. What that means is that you have a certain minimum length of sequence that you need to cover if you want to do analysis of a receptor gene. Now, 2 by 250 base pairs, you might be able to get away with that. If there is a very long CDR3, probably those particular sequences would be excluded. But for example, for most genes, particularly genes which don't have a D, the isotypes which don't use a D, you probably will get away with those. So light chains in the Ig locus, or maybe TCR alpha genes or TCR gamma genes, you can probably sequence those, but you might have a little bit of problems if you're doing genes which contain D genes or which have large CDR3s. So it could be possible to get away with that if you design the primers which are very, very close to the edges. But otherwise, I think you're sort of pushing your luck a little bit using a 2 by 250.
Thanks very much. The second question was, what do you recommend if we want to describe the Ig loci of a species where there are no references? Okay,
we have done that before with a couple of species. Obviously, we started with macaque where there was very limited reference sequences, but we have in our lab analyzed a couple of other species as well, and notably the guinea pig. Now, what we need to do initially is, of course, to create a library for the chain you're trying to amplify. If, for example, if you're doing the IGH locus, it's probably best to create a library of a more naive isotype, such as an IgM. Quite often, IgM, the constant region where you're going to be placing your primer, is kind of similar in a lot of species. So if you don't have any sequence from the genome of the species you want to characterize, if you look for the most closely related evolutionary species which has been sequenced across the IgM locus and design a primer based on that sequence, quite often that will work. Now, what you'll want to do at that point is to create a 5' race IgM library. That means you don't need to know any of the additional sequence. You only need the IgM sequence, which gives you the constant region cDNA synthesis primer. The rest of the amplification primers are based on the template switch that you get from their 5' race procedure. And if you get that working, then you can use a inference program, and probably you will need a starting database to actually begin the process. The starting database doesn't need to contain sequences which are accurate for that species. So you can use a reference database from a related species, and an inference program hopefully should be able to replace all of those sequences with germline sequences. You will have a problem in the sense that you don't know any numbers of genes that you're aiming for. So it's best that you try to do it in a way that doesn't limit the number of output sequence you get. So you can do that with IG Discover. We did that initially with the macaques, doing an iterative process. So essentially, the first round of gene discovery will find that the high frequency genes. It will replace the database, and you will be able to iterate the process again several times. So finding that the less frequently expressed germline sequences each time. At the same time, it will carry out a lot of filtering just to make sure that you're not pulling out false positives. Now, in the end, you will get a database which contains a lot of germline sequences, but you will not know whether these refer to allelic variants of the same gene or whether they are completely different gene sequences. That will probably require the integration of some degree of genomic information. But even so, the database will be useful if you intend to do assignment studies, for example, in an immunization project. You will be able to get accurate SHM information using that type of database. So hopefully that's useful information for you.
Yes, thank you very much. I think that added a lot. And we do have one final question. How do you compensate for sequencing errors due to long sequences? Oscar, that might have been directed to you.
Yeah, so we were really conscious about this, especially early on, because early on the long read sequencing platforms did generate reads that contained a lot of errors. But the most recent platforms, they're generating long reads that have minimal errors. I mean, there's still errors, but the accuracy of the reads are greater than 99.9% accurate. But we go even beyond that. So that's just the starting point. The starting point is 99.9% accurate. But in the assembly pipeline, there's a step where we take all the input reads, and then the assembly pipeline, what it does is it collapses similar reads to generate even more higher quality reads. So it's basically finding support of multiple. It's finding support by multiple reads and then generating a highly accurate consensus sequence. And then that highly accurate consensus sequence goes on to be used to create the assemblies. And so we kind of show that the initial error rates in the assemblies, even in the raw reads, even though they're still 99.9% accurate, they don't really affect our variant call set. And we tested that using ground truth samples and other orthogonal methods. So the end results, the allele calls and the VCF calls, I'm not going to say they're always 100% accurate, but almost all the times they're highly accurate.
Well, thank you very much. I do appreciate your efforts and contributions here. So in concluding, I'd like to thank our speakers for relating their insights into computational mining of immune receptor germline gene loci variation. And I'd like to thank our attendees for joining the webinar today. An on-demand version will be available in a few days. I'll send a link by email to everyone who registered. Please feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again and have a great rest of your day.